Hello, my name is Joshua Krupp. I'm a postdoctoral fellow in the laboratory of Joel Levine at the University of Toronto at Mississauga. In this video article, I'll be demonstrating a dissection technique that I developed for the isolation of the adult oenocytes from the fruit fly Drosophila melanogaster. In fruit flies, as in other insects, a waxy layer composed of hydrocarbon compounds coats the outer surface of the cuticle. This waxy layer provides resistance against desiccation and protects the animal against other environmental challenges. Several of these cuticular hydrocarbon compounds also serve as semiochemical signals. As such, they mediate pheromonal communications and influence behavior. The oenocytes, the cells of which I will be demonstrating how to dissect, are considered to be the primary site for the production of these cuticular hydrocarbon compounds. The dissected preparation demonstrated here will allow for the detailed molecular and genetic analysis of the oenocytes from an adult fruit fly. For this dissection procedure, some basic dissection tools are required. First, a Pasteur pipette is required for dispensing chilled insect media. Second, tungsten filament is needed for making a fine dissection needle. To produce this needle, the tungsten filament is first inserted through a fine bore hypodermic needle. This provides the tungsten filament with needed support. The tungsten is then passed slowly through a flame. This acts to burn off some of the tungsten at the tip, creating a very fine and very sharp needle. Next, a sill guard dissection plate is needed. Sill guard comes in several shades of gray, or it can be transparent. The sill guard provides a